So now we will discuss about the classification of bioremediation and different factors that affect the rate of bioremediation, right? So there are three main classes of bioremediation. These three classes are: number one is biotransformation, and the second is biodegradation, and number third is biomineralization. So, what is biotransformation? It's a little definition, a little introduction. Yeah, that is, it is the alternation of contaminant molecules into less or non-hazardous molecules. That is known as biotransformation. Or simply to transform one molecule into another less or non-hazardous molecules is known as biotransformation. Next is biodegradation. It is basically the breakdown of organic substances into smaller organic or inorganic molecules. This is known as biodegradation. Third one is mineralization. It is a complete biodegradation of organic materials into some inorganic constituents, such as carbon dioxide and water. So these are the three main classes of bioremediation, right? As we have discussed a lot about what is bioremediation, now we will see how does bioremediation work. What are then after that we will discuss some factors which affect the rate of bioremediation. So, in order for the bioremediation to take place, there should be some kind of microorganisms that should be present at that contaminated site. So, microorganisms, firstly, they should be active and healthy in order to perform the bioremediation. There are many bioremediation technologies that assist these microbes to grow and increase their population by creating optimum environmental conditions for them to basically detoxify maximum amount of contaminants that are present at the contaminated site. Okay. So the specific bioremediation technology used, it means the kind of technology which we will discuss in the upcoming lectures. These biotechnology technologies are determined by several factors. For example, the type of microbe present at the contaminated site, the site conditions of the contaminated site, the quantity and toxicity of the contaminants or the chemicals that are being uptaken by the microbe. So different microbes, as I have discussed in previous lectures, there are different microbes that degrade different types of compounds and they survive under different conditions, right? Like we have discussed about petroleum hydrocarbons, like petroleum compounds, they are different bacteria, different species of yeast, which can degrade the petroleum component. Likewise, there are different conditions for those microbes. They require specific pH for so that they can perform the process of bioremediation. They can live at specific temperatures, like some microbes can live. Some microbes are cold-loving microbes. Some are heat-loving microbes. Some are the microbes which can live at the moderate temperature, right? So there are some indigenous microbes, and indigenous microbes are basically those microorganisms that are found already living at the given site, right? The site where the contaminants are present, at that site, the microbes which are present, those are known as indigenous microbes, right? So to stimulate the growth of these indigenous microbes, there should be proper soil temperature, there should be supply of oxygen, there should be supply of nutrient contents, okay? And these are the requirements which are needed by the microbes to carry out the process of bioremediation, right? And if the biological activity needed to degrade a particular contaminant is not present in the soil at the sites, right? Means the microbes are not present at the contaminated site. We can basically use the microbes from other locations, okay, whose effectiveness has been tested by different processes, and then those microbes are added to that contaminated site. So these kind of microbes are known as exogenous microbes, right? So the soil conditions at the new site may need to be adjusted to ensure that the exogenous microbes will either survive at that site or not. Right? It is important. They should have proper. Their all requirements should be met so that they can perform exogenous microbe. Basically, inform the basically start or perform the process of bioremediation, right? So there are the microbes that can perform their activities or basically they need oxygen for performing their activity, right? Those microbes are known as aerobic microbes, whereas some microbes they don't require oxygen to survive, so that kind of microbes are known as anaerobic microbes. So it all depends what type of contaminant you are going to decontaminate, and for that contaminant, what type of microbe you are using, right? Either you are using aerobic microbe or anaerobic microbes. According to that, you will provide those requirements, or you will meet those requirements, or you will provide those conditions for the microbe to survive at that site and perform the process of bioremediation, right? Um, in aerobic conditions, microbes use available atmospheric oxygen in order to function. With sufficient amount of oxygen, the microbes will convert many organic contaminants into basically they use or organic contaminants in their system, and as a result they produce carbon dioxide and water. Whereas anaerobic condition, they supports the biological activity or the bioremediation in which no oxygen is present, so the microbes can break down chemical compounds in the soil to release the energy they need, right? They don't, in short, anaerobic microbes don't need oxygen to perform their function, right? Sometimes during anaerobic processes of breakdown, the original contaminants, the intermediate products that are less or equally or more toxic than the original contaminants are created, right? There are some processes in which the contaminants that are degraded and they use more toxic form of the product that is very dangerous as compared to original contaminants, right? So bioremediation can be used to clean up the contaminated soil or contaminated water. So as we have discussed previously, the bioremediation application falls into two broad categories, that is in situ bioremediation and ex situ bioremediation. Right? As you know, in situ bioremediation, obviously the bioremediation that is performed at the site where contaminant is present is known as in situ bioremediation, and ex situ bioremediation, obviously the contaminated soil or water that is excavated from that site and then on some other location, different processes are performed to accomplish the process of bioremediation, right? In ex situ bioremediation, it is a kind of expensive process because you have to dig up that soil which is contaminated, or you will excavate that contaminated soil, or you will pump the water from the underground, which we want to treat, right? So this is all about how does bioremediation works, the different requirements of microbes, either they are present on the site or either we will collect microbes from some other location, and then we will put those microbes at the contaminated soil or water. Then aerobic and anaerobic conditions, and then lastly, whether that bioremediation is going to be in situ bioremediation or that is going to be the ex situ bioremediation, right? So there are some factors of for bioremediation to take place. Basically, the control and optimization of bioremediation process is a complex process of many factors. So these factors include the existence of microbial population that is capable of degrading the pollutant and the availability of the contaminants to the microbial population, right? So microbes, as you know, they can be isolated from almost any environmental conditions, as I have discussed earlier. For example, some microbes can survive or grow at sub-zero temperature. That is extreme heat, or desert. Some microbes can live in extreme heat or desert conditions. Some microbes can live in water. Some need excess of oxygen. Some don't need oxygen at all, so that they can convert the contaminants or pollutants into less non-hazardous compounds, right? So microbes can be subdivided into two main groups, that is aerobic microbes and second-hand anaerobic microbes, right? So the microbes which need oxygen for their survival, or in this case we can say, in case of bioremediation, we can say the microbes which require oxygen to carry out the process of bioremediation are known as aerobic microbes, right? So aerobic, there are some examples of aerobic bacteria that have the degradative abilities, and these bacteria include Pseudomonas and Sulfangiomonas. These are the two examples of aerobic bacteria that have the degradative abilities, and obviously the aerobic bacteria, so they require oxygen so that they can carry out the process of bioremediation. In addition to these microbes, there are also some other bacteria, right? Rhodococcus and Mycobacterium. These are also aerobic bacteria, right? So these microbes have been reported to degrade pesticides, hydrocarbons, either they are alkanes or any other compounds. So basically many of these bacteria use the contaminants like pesticides and hydrocarbons as a source of carbon and energy, right? And as a result, what they will produce? They will produce carbon dioxide and water, right? These pesticides or these hydrocarbons or these alkane compounds are not harmful for these bacteria, as they have the degradative ability by the nature that they can take up those pesticides or those hydrocarbons in their system as a source of carbon and energy. And as a result, after degrading those harmful compounds, they basically release carbon dioxide and water into the environment, right? So there are two other microbes that are considered as aerobic microbes, which include methylotrophs and ligninolytic fungi. So firstly, we will discuss about little bit about methylotrophs. They are the aerobic bacteria that can grow utilizing methane for carbon and energy. So basically, these are the bacteria which use methane as a source of carbon and energy, right? So the initial enzyme in the pathway for aerobic degradation is methane monooxygenase, right? And this enzyme has a broad substrate range, and it is active against a wide range of compounds. These compounds include chlorinated aliphatic trichloroethylene and 1,2-dichloroethane, right? So it means these microbes use methane as a source of carbon and energy, but they have the enzyme. As you know, enzyme has active site on which the substrate will attach and perform its function, right? So these microbes has the enzyme that is methane monooxygenase, right? And it has broad substrate range. It means it will attach some kind of substrate on its active site so that it can perform the process of bioremediation. So those substrate includes chlorinated aliphatic trichloroethylene and 1,2-dichloroethane, which means that methylotrophs basically degrade chlorinated aliphatic trichloroethylene and 1,2-dichloroethane, right? They are using that those compounds, those contaminant as a substrate, and then the enzyme basically degrade these contaminants into non or less hazardous compounds, right? Next, ligninolytic fungi. So fungi such as white rot fungus, that is Phanerochaete chrysosporium, they have the ability to degrade an extremely diverse range of toxic environmental pollutants or persistent toxic environmental pollutants, right? Like methylotrophs, these fungi also need some kind of substrate to perform the function of bioremediation. So in the case of ligninolytic fungi, the substrates used include straw, sawdust, or corn cobs. These are the three substrates that are used by ligninolytic fungi so that they can degrade any toxic or persistent environmental pollutants from the environment, right? From the soil or from the water, right? As in the case of methylotrophs, I have discussed is जो भी pollutants और pollutants हैं, for example chlorinated aliphatic trichloroethylene है या 1,2-dichloroethylene है, they act as a substrate for the enzyme of these microbes, right? But in case of ligninolytic fungi, they need any other substrate to carry out the process of bioremediation, so that the enzyme of these ligninolytic fungi basically they attack that substrate on their active site, and that substrate is straw, sawdust, or corn cobs, right? And then they will do is they will degrade the any toxic environmental pollutant, right? They need additional substrate. In short, methylotrophs they use the pollutant or chemical as a substrate, but in case of ligninolytic fungi, they need additional substrate like straw, sawdust, or corn cobs to carry out the process of bioremediation, right? Next, anaerobic microbes, they obviously can survive in the absence of oxygen. Anaerobic bacteria are not frequently used as aerobic bacteria. These are less frequently used as compared to aerobic bacteria, right? So there is an increasing interest of scientists in anaerobic bacteria so that they can use those bacteria for the bioremediation of some polychlorinated biphenyls which are present in river sediments, or they can use these anaerobic bacteria for the dechlorination of some solvents like trichloroethylene and chloroform, right? So scientists or researchers are trying to use these anaerobic bacteria for the process of bioremediation. But mostly aerobic bacteria are used for that purpose, right? Next, there are some environmental factors that influence in situ bioremediation, right? We will discuss some techniques of in situ bioremediation of soil and water and ex situ bioremediation of soil and water in the coming lectures. But right now we will discuss some, before discussing that, we will discuss some environmental factors that basically affect the rate of in situ bioremediation, whether we are providing that factor to the microbe, either its bioremediation rate is increased or decreased, right? So its environmental factors include, number one is the pH, right? So most laboratory-based bioremediation studies have been carried out in the about neutral pH, since majority of the bacteria have optimal growth at the neutral pH range, right? In some studies, in some cases, there are some the microbes that can live in the acidic environment. So it all depends what type of microbe we will provide that kind of, or we will maintain that pH for the specific microbe. Either it is acid loving microbe or basic loving microbe, right? But majority of the bacteria which we use in the laboratory to carry out the process of bioremediation, those are mostly those basically live at neutral pH, right? Next factor is the temperature. So temperature directly affects the metabolism and growth of bacteria. There are vast majority of in situ bioremediation applications that have been carried out under mesophilic conditions, right? Like those microbes require 20 to 40 degrees centigrade temperature for their survival. However, there are some thermophilic or thermotroph species that are capable of degrading a vast variety of organic compounds that are present in contaminated soil or contaminated water. So they need highest temperature, right? 60 to 70 degrees centigrade, right? Likewise, there are some microbes that can live. They are cold-loving, basically microbes that can live. That they require lowest temperature to carry out the process of phytoremediation, right? So it all depends what type of microbe we are using. Uh, after studying many techniques of bioremediation, it has been proposed like most microbes they like the moderate temperature to grow, and that moderate temperature is required for the microbes to increase the rate of bioremediation, right? Next factor is the water content, right? As you know, water content in the soils and sediment is an important factor affecting the bioremediation rate, right? What type of a soil it is, it should have some kind of moisture in it, which microbes will use to carry out the process of bioremediation, which microbes will use to carry out the process of bioremediation. So we can say microbe generally require water activity values in order to metabolize and grow, right? Mostly water activity value of most microbes is 0.9 to 1. So the majority of bacteria they grow at these activity values, right, 0.9 to 1. Right? So to some extent, there should be availability of moisture in the soil, so that the microbes either those are the indigenous microbes or the exogenous microbes, they can carry out the process of bioremediation, right? Next one is soil type. Okay, it also depends if we are doing the in situ bioremediation, like the what type of soil it is. In situ bioremediation rates are enhanced when the soil is granular or porous, right? There's some kind of pores that are present in that soil. What is we can say. advantage of the porous soil, it means the microbes can easily get the oxygen from the air through those pores, right? So in that way, bioremediation rates are enhanced when soil is porous. So in situ degradation rates are slowed down under unfavorable geological characteristics, which include low permeability of soil, fractured rocks, or waterlogged conditions, right? Or some arid conditions. Under these conditions, the in situ bioremediation rates are slowed down, right? So it also depends whether the rate of bioremediation is increased or decreased. It depends what is the concentration of contaminants in that. Either the concentration of the contaminant is high or it is low, right? If we say the concentration of contaminant is more than 5% or 5% or more than 5%, then that soil is said to be have higher concentration of contaminants, right? For that purpose, the soil is agitated in a purifying water solution containing interface active agent and then separated from the oils. After that, bioremediation is started to efficiently clean up the soil, right? There is some kind of pre-treatment of that soil which contains higher level of contaminant, right? Like in a purifying water solution that has some kind of active agent and that will basically remove some of the contaminants from that highly concentrated soil, right? Then after that, when the concentration of contaminant became low by this pre-treatment, bioremediation started efficiently to clean up that soil, right? Then that work or that bioremediation is continued to make this process even more efficient and more effective. Next one is if the contaminant that are present in the soil, they have a lower concentration, right? Obviously, they have a less than 5% contaminant present. We can say that soil have a lower concentrations of contaminant. So obviously, they don't need any kind of pre-treatment like higher concentration of contaminants. They are straight away subjected to the bioremediation process, right? So it takes about 6 months to a year to purify soil containing 2% of heavy oils, right? 2% obviously it means it have lower concentration. So it is very time-taking process. Obviously, 6 months to 1 year is needed to purify the soil that contains 2% heavy oils. But if the concentration of these contaminants is only 0.8%, this job of remediation can be done in about 1 to 2 months, right? So it all depends. Concentration is whether lower, or the lowest, right? Whenever the concentration is low, the less time it takes to bioremediate or to clean up that contaminated soil, right? So this environment-friendly method make it possible to recycle and to reuse that soil that is basically decontaminated by using bioremediation process, right? Next factor is the nutrient availability. So in almost all cases, nutrient supplements like addition of nitrogen and phosphorus, it significantly increases the bioremediation rate, right? So why there's a need of nutrients? Obviously, microbes are there present in the contaminated site, or we are introducing the exogenous microbes. Obviously, they need some kind of nutrients so that they can survive, they can increase their population so that bioremediation can be carried out, right? If we provide more nutrients, there is a more microbial growth. As a result, more population, it will perform the process of bioremediation at increased rates, right? So It has shown very good results in hydrocarbon bioremediation of soils and groundwaters when nitrogen and phosphorus levels have been shown to be limiting. Right? So carbon is a basic element of living form, so it is needed in greater quantities than any other element. In addition to hydrogen, obviously we are providing hydrogen, we are providing oxygen, we are providing nitrogen. Carbon obviously constitutes 95% of the weight, right? So the nutritional requirement of carbon to nitrogen ratio is 10 ratio 1, right? If there is 10% carbon, there should be 1% nitrogen. And the ratio between carbon to phosphorus is 30 ratio 1, means if there is 1% phosphorus, there should be 30% carbon. So these nutrients are required for microbe to increase population and enhance the process of bioremediation. And obviously, its example is like if I gave a task to only one student, any task, I asked that student to make an assignment individually. That will take time as compared to I just make the groups like I make a group of 5 to 6 students, and then I assign assignment to them, and then what they will do, they will distribute the subtopics of the topic, and then that one student which is doing the assignment individually in the 3 or 4 days, these the group of the students that will basically complete their assignment in just 1 day or some 2 to 3 hours, right? So same is the case with the microbes. Like if we want to increase the population of microbes so that they can efficiently, in the form of groups, they can perform the process of bioremediation. We will basically provide them with the proper pH, with the proper temperature. Most importantly, the nutrient availability, right? Next, very important factor is external electron availability. So there is a need of electron for these microbes to carry out the process of bioremediation. Like in many in situ bioremediation techniques, provision of oxygen supply is needed to enhance the aerobic respiratory breakdown of organic contaminants, right? So in that case, addition of hydrogen peroxide is used to introduce oxygen, right? There are some cases like if microbes are living in a porous soil, what they will do is they will get oxygen from the air by the help of the pores that are present in the soil, right? But there are some microbes that are present in non-porous soil. So we have to provide them basically oxygen so that they can live in that environment, grow in that environment, and carry out the process of bioremediation. So basically, external electron availability is how we provide oxygen to microbes that are living in some kind of environment, like they can't get the air or oxygen from directly from the environment. So we basically add hydrogen peroxide in that system or in that contaminated soil. And basically when hydrogen peroxide is broken down, it is, as you know, it is broken down into water and oxygen. So basically microbes will get water as well as oxygen from that hydrogen peroxide, right? So hydrogen peroxide is about 70 times more soluble in water than oxygen, and its decomposition in soil yields zero point 0.5 moles of oxygen per molecule of hydrogen peroxide introduced to the contaminated site, right? As you know, oxygen is also soluble in water, but hydrogen, if we talk about hydrogen peroxide, which is a source of electron availability or which is a source of oxygen, this hydrogen peroxide is about 70 times more soluble in water, right? And if there is a decomposition of this hydrogen peroxide, it will give yield of 0.5 moles of oxygen per molecule, right? So this was about the external electron availability, right? In some cases, when we will discuss techniques, we will see that we will directly provide air from the environment, basically provide air to the microbes that are living in the contaminated soil from directly from the air. So in some cases, we will use hydrogen peroxide as a source of oxygen, right? We will just sprinkle the hydrogen peroxide, or we will inject a hydrogen peroxide in that contaminated soil, so that microbes can take up that oxygen and carry out or enhance the rate of bioremediation, right? So the next factor is bioavailability of organic pollutants. This is very important factor that is governing the rate of in situ bioremediation, right? Improvements have been made in bioremediation rates by addition of some biosurfactant or synthetic detergents to the contaminated zone. For example, there are some biodegradable solvents which assist in desorption and dissolution rates, also exhibit increase in the biodegradation of some adsorbed pollutants, like adsorption of some pollutants by the soil particles, right? So basically, these organic pollutants are required by microbes, so they can carry out the process of bioremediation more efficiently. As we have discussed in the topic of, I guess, bioremediation of marine oil spills, that there is a need of some oleophilic fertilizers, and what those oleophilic fertilizers will do is they will just they are used for dissolving oil components or petroleum based components into the water, right? So likewise, there are some synthetic detergents, or there are some biosurfactants that are needed by microbes, so that they can carry out the process of bioremediation. Or in other sense, we can say we can enhance the rate of bioremediation by using those organic pollutants, right? Next factor is co-metabolism. So what is co-metabolism? As we have discussed in case of ligninolytic fungi, that they need additional substrate to carry out the process of bioremediation. Like ligninolytic fungi, they need substrate like corn cobs, sawdust, or straw to carry out the process of bioremediation. So this type of so that microbes can do the process of metabolism, and then they can convert that toxic contaminant into non or less hazardous form, right? So this type of metabolism is known as co-metabolism. If additional substrate is required by microbe to carry out the process of bioremediation, that is known as co-metabolism, right? In some cases, like in methylotrophs, there is no need of substrate, like the pollutant or the contaminant is behaving as a substrate to carry out the process of metabolism in the microbe, so that they can convert that contaminant into less hazardous form. But in some cases, like in ligninolytic fungi, there is a need of additional substrate. Obviously, they will do the bioremediation of that pollutant or contaminant, but there is a need of extra substrate that will attach with the active side of the enzyme of the microbe, so that they can carry out the process of bioremediation, right? So the bacterial transformation of DDTs, PCBs, these provide examples of both aerobic and anaerobic co-metabolic biodegradation, right? So rate of bioremediation can be enhanced by the process of co-metabolism, right? Next factor is the gene expression. Obviously, as I have told you earlier, if microbes are taking up the particular contaminant, there are the genes that encode for specific enzymes, and that enzymes are basically involved in the biodegradation of that pollutant or that contaminant, right? Like in case of methylotrophs bacteria. They are the pollutant or contaminant are behaving as a substrate, and as a result, those methylotrophs are converting that pollutant or contaminant into less hazardous form, right? So there is a need of gene expression, right? If gene is not expressed, the microbes won't be able to carry out the process of bioremediation. Which gene, obviously, whenever we introduce any contaminant or pollutant into the microbe, or we can say in the plants, in case of phytoremediation, there is a need of gene expression of that particular gene which will encode specific enzyme, and that enzyme will actually attach that contaminant or pollutant at its active site, and then it will convert into less hazardous forms, less hazardous compound, or that will convert into carbon dioxide or water, right? Or in other case, like in ligninolytic fungi, obviously there is involvement of enzyme, and enzyme, how obviously enzyme will start its function. Obviously, the gene had to express itself, right? We can say that there is a microbe, for instance, there is a microbe that can carry out the process of bioremediation, or which type of contaminant we can say, like petroleum hydrocarbon. So there should be some gene in that microbe which is responsible for expression, for the expression of that gene. How that gene will express, it will encode for some specific enzyme, and that enzyme is going to, that enzyme of microbe or that enzyme of plant is going to basically degrade that contaminant, right? So if genes are not expressed, bioremediation cannot be carried out, right? So these are some of the factors which are important if we are doing the in situ bioremediation, right? Like these all nine factors basically, either they increase the rate of bioremediation, or they enhance the rate of bioremediation, or they slow down the rate of bioremediation. It all depends upon these nine factors, right? If we are providing all the conditions, all the factors to the microbes which are living in situ, we can obviously enhance or increase the bioremediation process. And obviously it is needed in case of in situ bioremediation, because it's a very, it is done on a very large scale. It's a very time-taking process. So if we want to enhance that process, so that the process which is taking 6 months or a year, we try to complete that process in some 2 to 3 months or 1 to 2 months. We just basically provide all these factors to the microbe so that they can carry on the process of bioremediation, right? So this was all about today's topic.